Okay, so for today's video, we're going to describe the molecular characteristics of the contractile filaments found within the functional unit of a skeletal muscle, the sarcomere. Describe the mechanism by which an action potential in the sarcomere results in release of calcium from the sarcoplasmic reticulum, followed by a description of the molecular mechanisms of muscle contraction. In part two, we're going to discuss the various sources of energy utilized for muscle contraction, include the relative amount of each source available to the cell, the advantages and disadvantages of each type, and what conditions each might be used. And then we're going to describe the two main types of smooth muscle, describe the molecular mechanisms of the smooth muscle contraction, smooth muscle. And then we'll begin at calcium entry and at the point of contraction ends. And then we'll compare and contrast smooth skeletal muscle structurally, functionally, and regulation. Finally, we'll describe and explain, and then compare and contrast the muscle sensory receptors, muscle spindles, and Golgi tendon organs, and describe the stretch and reflex and withdrawal reflex. Okay, so we'll start off. So with our sarcomere, we have an alpha neuron right there. That's our muscle fiber running right there. So I'm just going to draw this out first. Just because, why not? So, here I have alpha neuron coming on, and here we have an N2 receptor. We're going ahead. Okay. So, we're going to draw a Z disc. So yeah, so we have jeez. Okay, so this whole unit we call a Z disc. A whole bunch of these will make up our sarcomere. So the Z disc. This area right here is titin, and here we have nebulin. So along titin, we have our myosin light and heavy body chains. Where's red? Along titin, we have this, So in red here we have myosin, which has two parts of it. Head's got a tail. And heavy chain, light chain. So on the heavy chain we have the heads. The light chain would be the tails. Heavy and light chain. So the light chains it controls the myosin. And then the heavy chains, those will bind onto a molecule during muscle contraction called F-actin. Which I'm going to label in green. It goes helically around our tubulin. It's called, the individual subunits are called G-actin. Then we put them in their little helical shape, we call them F-actin. G-actin. Okay, so we'll go around there like that. So, yeah. So the main players that we're going to be looking at, myosin and actin. Like I was saying earlier, globular G-actin will form an entire chain of monomers that are polymerized into filamentous or F-actin strands. And then there's an association between regular proteins of tropomyosin and troponin. Troponin is bound around the F-actin sites. Looks like Mickey Mouse ears, where it has a T, an I, and a C. And what we see with muscle cells is that they're more easily excitable because they're more negative at the resting membrane potential. So calcium, unlike what we saw with the nerve cells, how potassium was the major player of helping them do their thing. In this case, it's calcium. So on the troponin C unit, it has a high and a low affinity binding site. The high affinity binding site will always be filled with calcium, and that's what keeps the troponin unit together. The low affinity binding site will move out of the way a molecule called tropomyosin, which will block off this binding site for so yeah, it will, let's go back a little bit. So with calcium, that's a little bit different. We got the ECF, ICF. So a skeletal muscle, there is 100% is in the ICF, sorry, from the sarcoplasmic endoplasmic reticulum. We'll talk about that a little bit later. We're going through the actual action potential through it. Heart muscle, it's kind of unique. It's going to be 90% from the ICF, 10% for the ECF, because if it was 100% like it was a skeletal muscle, then there's a chance that you could potentially run out of calcium. We don't really want our heart to fatigue, because that's bad. Then smooth muscle, it's unique. With smooth muscle, we'll be taking 50% from the ECF and 50% from the ICF. Because the smooth muscle can do funny things. It can go in a phasic or tonic zone, but we'll talk more about that a little bit later. So yeah, it is fun. So troponin is going, well, yeah. So on the F-actin in green, sorry, I'm just putting down all the players so then I go through the action potential. It makes more sense. There's gonna be these little binding sites, and that's the myosin binding site. That's where myosin will bind onto the F-actin, and our troponin with its little Mickey Mouse ears is bound here, but there's also this other molecule called tropomyosin, which I'll use orange for. I don't want to blend these colors, but it looks like it's gonna be inevitable. Tropomyosin is bound around in that zone, and tropomyosin blocks the myosin heads from binding to the F-actin, which in later, that's one of the ways that we get muscles to move, is that binding in those two sites. So the I site of the troponin will be binding to the G-actin, so that's our I. The T will bind into the tropomyosin, so these two are bound. Sorry, that picture's ugly. And then the C, like we were saying earlier with the high affinity and low affinity. The high affinity will be bound first, and that keeps those 3 subunits together. And then the low affinity will change things with the muscle fiber being excited. So the low, once it gets bound, it'll cause the troponin to shove the tropomyosin out of the way. And that being bound out of the way will allow for our myosin heads to bind onto the F actin and make a really cool subunit that will do some fun stuff. So that is a lot of our players. For each tropomyosin we have 1 troponin unit that will shove it out of the way so then the myosin can bind. But next we'll kind of be going through so next part of it, let's go back to what our prompts are. Describe the mechanism by which action. Okay, cool. So moving on. So saying that a lot. Let's erase some of this stuff. Where's my eraser? There it is. Okay, so excuse me. It's gonna be a late night. So we have right here what I drew out was called a motor unit. This is just one muscle fiber, but this alpha neuron will innervate a lot of different other neurons and fibers. So yeah, could be different variations. Could be a 1-to-1, 1-to-10. For smaller muscles like the eyes, it's usually a 1-to-1 ratio of alpha neuron to number of fibers that it innervates. For larger muscles, it'd be like 1-to-1,000. Also with small muscles, it's like 1-to-10. So the big muscles, like we're talking the quadriceps and those kind of larger, put in heavier work muscles. So with our action potential and sarcolemma, so the initiator will get a bong, our spirit influence that will go down. Our calcium will come in through V-gated from the previous signal. Calcium will enter, causing vesicles to release acetylcholine. It'll bind to the receptors on the motor plate, causing depolarization. So we'll release acetylcholine after the calcium binds to snare proteins, causing the vesicles to release acetylcholine into the synaptic space. Then here we have an N2 receptor. Those will bind to acetylcholine. And then the action potential then propagate along here where we have a voltage gated sodium channel or potassium channel. And that sodium voltage gated potassium channel so that'll release sodium, potassium to head down in this area right here, which we call a T-tubule. T-tubule. So the T-tubule will get filled with potassium and sodium, and that'll cause a depolarization. It'll reach a certain threshold that this DHP channel will which is a voltage gated, it's a specialized voltage gated calcium channel, which is connected to this ryanine receptor channel. And when the voltage enters and it reaches a certain spot, that will cause these two to open, and then our sarcoplasmic, this is like a cap, it just causes a change. Then this can open, and we can leak out our calcium that's high in here into the sarcoplasm into here, which here we have a bunch of those little Z discs that will be drawn up later. But yeah, so going through we want to release calcium in the sarcoplasm, so then once calcium binds to those areas, like we were saying earlier, calcium will bind to the C, inducing conformational change of tropomyosin out of the way, and then tropomyosin out of the way of blocking the myosin binding sites on the F-actin. And once those bind to the exposed actin sites, it'll form a cross bridge, and it'll undergo the power stroke cycle, which will, ATP hydrolysis will energize the myosin binds the actin, it'll release ADP and inorganic phosphate to generate force. Heat will be released at that same time. And then when the calcium is actively pumped back into the SCR, because we have a circuit pump, ATPase pump, that will take the calcium from the sarcoplasm back into the SCR, and it uses that with ATP, which is pretty nifty. So yeah. It's pretty good. So with that, we can go into the power stroke cycle. So I'm just gonna draw half of the Z disc. So we have it right there. And then we have ATP bound there, inorganic phosphate, right there, ADP. Okay, so this is our myosin head. It's phosphorylated. I'm going to introduce calcium into the system, which introducing calcium will then be bound to the tropomyosin. Tropomyosin will then expose the P-actin. So expose the myosin binding site. So it'll look something like this. The head will be bound, but we still have the phosphate connected. And by doing this, we'll release some heat. Heat will come off. And then from here, we will release our ADP, which is that right there. I just want to draw it again. We'll also release phosphate, the inorganic phosphate. And this right here is called a power stroke. We're going from a cocked to an uncocked position, so there's a release of energy that causes the muscle to move. So in that position, the myosin head is what we call uncocked. From here, we'll then the myosin head has ATPase or ATP kinase on it, so we can introduce ATP back into the system. And then introducing ATP back in the system, we put the ATP onto the myosin head. We're still uncocked, so the ATP will go back onto the head like it was before, and then to be able to go back to our original position, we will hydrolyze the ATP, so it's back into ADP and inorganic phosphate. The ADP is just kind of floating around once we kick it off. We don't fully understand, I think, where or what happens to it, but it's there. So that's our power stroke in muscle cells, and we call this the cross bridge cycle. So yeah, this is the mechanism of how muscle cells move. We'll have a bunch of these Z discs within a muscle cell, and they'll independently move it closer together, so that they actually kind of move the muscle fibers further in. Because if you thought about it, or like a rope, you want to be able to maintain tension the entire time. Otherwise, you're not able to move the muscles quickly. But I mean, that'll be a little bit further on when we're talking more about the Zeke apps and whatever. So yeah, to be able to get calcium back in, like we were saying, it's via the circuit pump right there. Uses ATP, which does cost energy. So it takes a little bit longer to go through it. So yeah, that's the cross bridge cycle. Next, I think we're going to be talking, well, let's do this. So if we were to take a cross-section analysis of, that's my muscle cell. So here in the red, we'll have actin. And here in the blue, or sorry, red is going to be myosin. And then here in the blue, let's drop these. Blue, we have actin. So getting like a pump in the gym is we're growing the cells to make the cells bigger. We call that hypertrophy. But hyperplasia is to be able to make actual more cells. Hypertrophy is just one and done. It's the muscle cells lysing, and then they'll heal themselves to actually get bigger. To make more muscle cells takes longer, and we call that hyperplasia. So yeah. Next, we can talk about fatigue. So when we have this sodium and potassium going in, with fatigue, we have two flavors. We have, so here, fatigue. So we have one type, which is psychological, which it's your brain telling you like, hey, we're done. We don't want to do anymore. You can't overcome it. It's also called central fatigue. Another name for it. This is the mind over matter. But eventually, mind can only do so much. It can do a lot, but only so much. We get into physiological fatigue. It comes in two flavors. It's either low frequency or high frequency. With the physiological low or high frequency. With a low frequency, it's where the circuit pump becomes saturated. And we're not able to pump calcium back into, with the circuit pump, we'll build up magnesium, and the magnesium will get in the way of the calcium getting back into the sarcoplasmic endoreticulum. So with the ryanocine channel, less will be released. Yeah, so we'll get build up the magnesium there. Less is going to be released with the magnesium entering the RYR channel. So because of that, we see that low frequency with a high frequency. If there's too much of a buildup at charge in here, the muscle cells aren't able to depolarize, and there's a less chance. The calcium will have a less drive to want to leave the cell, if I remember right. So to be able to, So if it gets too high, and that there's always gets an influx of calcium, it leads the muscle to not work. So we have these chloride leak channels and chloride voltage gated channels that will cause chloride to enter the cell to balance it back out, so that this has a balance of charge. So we don't, but that does take some time. So that's kind of like when your muscles you're lifting really heavy and then the muscles give out, and then you wait a second and then you can be able to do it again, is because of the cell trying to get back to where it's supposed to be. It's obvious we need ATP to be able to do the cross bridge cycle, cross bridge cycle. So the cell gets it from, or these muscle cells, and also depending on what type of cell it is, gets it from different places. So let's start there. Actually, before there, we'll go through some different types of muscle fibers, because they all do use different sources of ATP uniquely and unique to their type. So, with these muscle fiber types, the skeletal muscle fiber types, muscle fibers and fiber types of skeletal muscle. Skeletal muscle come in 3 flavors. So we have type 1, we have type 2A, and then type 2X. In humans and animals, they call it type 2, or type 2B. So with ATPase activity, for our type 1, it's going to be slow. ATPase activity. For a type 2A, it's going to be medium. And here it's high. I mean, lots of energy for a type 2X. Usually for a type 2B, we're going to be using cytolytic ATP, or cytosolic, sorry. Cytosolic ATP, or creatine phosphate. For medium, it depends. It can either go anaerobic or aerobic. Our type 2A muscle fibers. So yeah, if we're going aerobic, we'll do some glycolysis anaerobic. The anaerobic is, so we can do aerobic cellular respiration, Anaerobic is typically favored with type 2A, and when we do that, we'll use glycolysis, which is where we break down glucose into some amount of ATP. And then for type 2 or type 1, typically use an aerobic cellular respiration to get energy. For the fatigue, for our legs, so we got fatigue, type 1A, or we can kind of think of it kind of like our legs, or this is more legs, but it's non-fatiguing like the muscles in our back, and our legs too. It would be very, if you're like just standing, you can kind of stand indefinitely and your legs don't get tired. For type 1A, it's fatigue resistant. And type 2X, it's pretty easy to fatigue them. Fatigable, that's what we're going to call it. So for mitochondria, because we're mainly going to be using aerobic for type 1, there's lots of them, medium, and then few. For our blood supply, mitochondria, blood supply. For here, there's quite a bit. It's high, which leads to it having a red color. For here, it's medium with our blood supply. So it's still red, more pinkish, but mainly red. And here it's low with our blood supply. So these are actually white muscle fibers. And the reason why we have this difference in the color is because of the amount of myoglobin within these molecules, within these different types of fibers. So with all these different things, we kind of went or hinted at cytosolic and creatine phosphate and all whatnot. So creatine phosphate has this structure on it, PO4. Looks more or less like this with creatine phosphate. So this group makes it very similar to ATP, where this is a very high energy molecule, where breaking that bond will release a lot of energy and allow red blood cells to do work. The first source and main source for the type 2X is our cytosolic ATP, which on average it can last about 5 seconds. Creatine phosphate is the next one. Which can last a little bit longer at 10 seconds. Then we have via glycolysis, it can last approximately 10 minutes. Oh, a minute. And then we have aerobic cellular respiration, which will last for hours. So, like we were saying earlier, type 1 muscle fibers, they can last for really long periods of time because they're using aerobic cellular respiration. Again, 38 ATP per each one of their mitochondria, and they have tons of mitochondria, so they can keep going through that cross-breed cycle quite a bit and don't fatigue as easily, while these other ones, they run out of ATP. This is the only one that's under aerobic conditions. This is all anaerobic. And what I mean by that is these can operate without the presence of molecular oxygen, while this one, molecular oxygen is necessary for the last electron donating molecule. So yeah. So here we go. These two, we have an immediate energy source. Advantage is rapid ATP generation. Disadvantage is there is limited storage. It's also used in short, high-intensity activities. So with our cytosolic, we're doing type 2X muscle fibers, which are typically associated with like bursts of energy, like sprints and whatnot. Your sprinters, your linemen, or some dude driving down the court as fast as you can in basketball. Just high intensity activities overall. With our anaerobic glycolysis, the breakdown of glucose to lactate without oxygen is what we're doing there with glycolysis, kind of like fermentation or lactic acid fermentation. The advantages is we also are getting quick ATP production. Disadvantage is the lactic accumulation can lead to fatigue, and that's used in kind of more moderate activities, kind of like your mid-distance sprinters. Sorry, that's annoying. Like a 400-meter run, that kind of thing. And then with aerobic, that can last for hours. So that's your marathon runners going, your Ironman people, where they're trying to last for as long as possible. So now we can go with our different types of smooth muscle, I believe is the next place that we're going. So all different tubes like our arteries, whatnot, have smooth muscle around our eyes. Around our organs have smooth muscle that will be able to do kind of different stuff. Skeletal muscle is unique for it has striations inside of it, so it kind of gets those jagged patterns underneath the microscope. Smooth muscle does not have that. So our type of smooth muscle that we have single unit smooth muscle, and then multi-unit smooth muscle. Where the single unit, they all move kind of one singular unit, like our intestines, or in females, a uterus. And then with the multi-unit smooth muscle, we kind of see that independent functioning fibers, kind of like our erector pili, or the muscles around our iris. So smooth muscle cross bridge is similar, but also different. We can go into two different phases, or it can, So there's phasic and tonic with smooth muscle. So we'll draw out those cross bridge cycles. It's super similar in skeletal muscle, but there's some slight nuances. So we have a myosin unit, ATP, phosphate, all that right here. We're going to introduce calcium into the into the system, but then we'll also introduce this new molecule called MLCK. It's myosin light chain kinase. Will be added in MLCK. And then we will also be immediately adding in ATP. So let me draw that out. I'm going to mark it in this other ATP. Where's my blue? There's my blue. We'll mark that other phosphate being added on, indicating it there in blue. And our myosin won't bind if the other phosphate is not added. So if this blue one is not connected to this unit, then our myosin won't bind there. Next, we're going to go through our next stage of it, where we'll lose one of the phosphates and the ADP, just like we were doing in the other version of it. We'll have our power stroke, but the unique thing is that that other phosphate will remain on there. That's important. We'll then use our phosphokinase, add ATP into the mix. So we're still bound. phosphate's still there. But something that's unique with muscle cells is that we can just go straight in to hydrolyze the ATP. So we can maintain contraction for longer periods of time without having to go into being what's it called? Fatiguing as easily. So something that will regulate, or we'll talk about MLCK and the other ones here in a second. But to be able to go back to our starting phase there and there, we'll activate this molecule called MLCP. And a phosphate. Or sorry. Yeah, so we'll do MLCP to take off that blue phosphate. So yeah, phasic, that's what it looks like. The regulation of this MLCK is that when calcium will enter either the ligand gated or the voltage gated tails, we have calmodulin. It looks like a snake. When it's inactivated, it looks like its tail is eating itself, and it gets activated when calmodulin will bind. And calmodulin binds, or sorry, we have this calmodulin molecule that's over here. So if it's inactivated, it looks like that. Once it's activated and calcium binds the calmodulin, then it'll look like this. And activation of it, well, yeah, it looked kind of silly there, but the calmodulin will be bound and is activated when calcium will bind to it. And we'll take a mole of ATP to phosphorylate the myosin head with that whole situation. So the advantage of phasic. So with our smooth muscle, we don't want to be thinking about it, because with skeletal muscle, it's an action potential that's sent from our brain. Well, not all of it. There's some reflexes that are obviously not controlled by our brain, it's controlled by our spinal cord. But we don't want to be constantly thinking like, oh, I need to relax, or I need to flex my tubes in my arteries. So this regulation is done involuntary, and it's also sustained. The regulation on skeletal muscle will depend on neural input, while smooth muscle, it's modulated by autonomic nerves, hormones, and local factors. One of those local factors we can see here is our MLCK and calmodulin. We'll draw out this other one, the MLCP. So MLCP kind of looks like a Pac-Man. And it has a phosphate right there. So this is its inactive state. So if if a phosphate's not added on rho kinase, then it won't work. So yeah, there's rho kinase. They'll add on that phosphate to the MLCP. And then let's draw him active. So if rho kinase does not add its phosphate, then the MLCP will be activated, like in this drawing over here. And the way to activate the MLCP is to have a sustained amount of calcium in the system. So if we have a sustained amount of calcium, then the muscle will go more into this tonic state, which we'll draw out that whole chart now. So we got our thing, it's doing its stuff. So similar with the tonic stage, we'll add in calcium. We'll add in the MLCK. We'll add in our ATP. That's not APP, that's not a thing. ATP, which that will then get us to here, where, again, very similar to the other one. From here, we'll kick off our phosphate like we did before. Kick off our ADP. But what's unique about the tonic stage is that we're going to be activating MLCK at MLCE, sorry, at this point. And I'll be kicking off that other phosphate. So in our power stroke, we'll look like this. Kind of more similar to skeletal muscle. We'll add in our ATP, like usual. And the regulation of ATP is going to be kind of binding and unbinding in this state right here. Once we bind it, so it'll kind of be falling on and off. And here what's unique with tonic is we have what's called the latch stage. That latch stage is kind of what our sphincters are doing. As long as we have that sustained amount of calcium, then we can go tonic and be able to get that sustained amount of calcium in the tonic stage, or with, the tonic phase of smooth muscle and its ability to do that. With latch is usually with our sphincters. And anytime that we're uncocking, that's our power stroke. So the difference in MLCP, it's early with tonic, but it's late with phasic. And we already talked about the enzyme regulation. Within phasic, we usually see that the MLCP is turned off. And we also see that probably the myosin head's binding is higher. Tonic, the opposite is true. And then we also find where the calcium will come from. So if we have this sustained amount of calcium for tonic, we're looking to try to get it from different G proteins. These G proteins will cause IP3, which will trigger our muscle cells to release calcium from there. Another way that we can get it from the ECF is from DHP voltage, kind of like we're doing with the muscle cells. Stretch activated is another way to release calcium from the ECF to the ICF. So those are just some of the ways to be able to get calcium in. So yeah, that's smooth muscle. So yeah, so this formation of the latch bridge of going from the unphosphorylated to the phosphorylated, and then once we're from that phosphorylated state, then we have latch right here with this blue arrow until we're hydrolyzed. And that latch stage is what keeps our sphincters nice and tight. Let's move on to the next section. Okay, so in this part we're going to be talking about the muscle spindles and the Golgi tendon organs. So to be able to do that, we will break it up with, so yeah, our muscle spindles. So muscle spindles detect stretch and initiate the stretch reflex to maintain muscle tone. So detect stretch to maintain muscle. Yeah, so detect stretch to maintain muscle tone. Earlier we were saying when muscles, the Z disc will move inwards or outwards depending on which way you're talking and which muscle in relationship to the other one. So these muscle spindles, if we're, say this is a rope, if we're moving it to be able to do it, this will sag and then eventually it latches on. But instead, we're going to move it closer with those muscle spindles to maintain the tension so that there's not a lag time or a severe lag time aside from the calcium causing the tropomyosin to get out of the way, so that, or calcium binding to troponin so that the tropomyosin can get out of the way so the myosin heads can bind to the actin and we're ready to start the cross bridge cycle. There'll also be a moment of waiting for the muscle fibers to get taut so then you can be able to move them. Muscle spindles solve that problem. And we also have the Golgi tendon organs. So Golgi tendon organs will detect tension and trigger inhibitory reflex to prevent excessive force. So your muscles, especially in your quads and your shins, are strong enough to. Muscles are definitely a lot stronger than bone, so you can potentially rip off your what's it called? You could rip off your bone with the strength of your muscles. So with your Golgi tendon, kind of doing things out of order, but it's okay. So we got your brain. And then somewhere along the way we have your leg. Not a great leg. So there'll be a signal that is from here, back up to your brain. Let's erase that. I don't like it. Plus, we're gonna do a different section because, well, not a different section, but let's talk about this other stuff first. So yeah, Golgi tendon organs detect tension. And will send an inhibitory response to excessive force. Okay. So within our brains, we have. Okay, so here is our brain. In some section of it, it'll send down a signal that will synapse somewhere in the spinal cord, and then the alpha neuron will go off into a neural unit. There's all these are muscle tissue, these are muscle fibers. So this is our alpha unit, and I'll branch off to innervate all these different muscle fibers so they can do their own thing. Yeah, so muscle fibers is what we have right here, or extrafusal fibers. Each one of these individual muscle cells will have extrafusal fibers intrafusal fibers within them, which are sensory fibers. We'll draw that in blue. Because running along with the motor neuron, we also have sensory neurons that will go from the muscle cell back up to the brain, So there, and I'll go back to where we're synapsing. There in green, we have our sensory. With our sensory fibers, they come in 3 different flavors. The first is type 1A, second is type 2, third is type 2B. So all 3 will return to the spinal cord on the same pathway as the neuron. So with the intrafusal fibers, we have muscle spindles, and then we have a bag spindle. So they're 2 separate things. Their muscle spindle apparatus, it's intrafusal fibers. It has a bunch of intrafusal fibers found within it. So their spindle will be like that. And we'll have the spindle apparatus is inside. We'll have one that's kind of short, and that's a little bit longer. Within these intral fibers, chains and bags within the spindle apparatus. The chain it's not used to generate force, but it can shorten. So blowing up that left one, here we have the chain. And then there we have a bag. It looks more like a kidney, but that's our bag. So coming in a signal, we have a gamma motor neuron that will synapse onto there. And then there's also some type 2 motor neurons that accumulate in between those little things. So yeah. With the bag, on the other hand, we can see that there's So with the chain, there's a large accumulation of the Z discs throughout, with a small section with the sensory neurons. Well, the bag's opposite. On the ends, we have a large distribution of the Z discs, with a lot more of the sensory going around within it, like that, wrapping around. So yeah. With the chain, it's more of a static gamma neuron, and then with the bag, it's a dynamic gamma neuron. So the static would, it can't sense as we're moving, it's just whether stretched or not stretched. While with the bag, it can sense, oh, we're stretching, oh, we're contracting. So with a type 1, type 2 says you're stretching. Type 1A is typically the more sensitive. So in a longer duration, or sorry, the type 2 has more to do with the duration of the stretch, while with type 1A, it's telling you the rates of like how much you're stretching and for how far. So how it works with the spinal cord with the stretch reflex, example you could do is the knee jerk. So right on your patellar tendon, has some of those sensory fibers. If you're hanging it loosely and if you're not thinking about it, you can hit. Right there will cause a signal of an EPSP to tell your quad to relax. Yes. So within your brain, you'll have different signals. One will be telling. Well, yeah, let's show how it works within the spine. So let's erase our bags and stuff. So more so with our reflexes, these aren't all. There you go. Okay, so here we have our spinal. This is a cross section of our vertebrae. It's not great, but you know, it'll work. So we'll have an incoming sensory outgoing motor. So, if we were to have like an agonist to the biceps to be right here, cause an IPSP to tell biceps to flex. Antagonist would shortly be close by, but to be able to cheat it, we'll have an short IPSP and make a new neuron. That once it's telling the biceps contract, it's telling the triceps to relax. So we have that interrelationship between the two. All alpha neurons will be doing stuff like that. So then as one muscle is contracting, the other one's relaxing, and vice versa for most muscles. So if we're talking about so we have the stretch reflex. Let's draw that one out. So we'll have our brain. So let's draw out our leg first. Here. Put in some muscle. Okay. So here we have one. This is our spinal cord. The reason why we have these reflexes directed to our spinal cord is the more that we can share the load of executive function away from the brain, and not executive function, but this kind of stuff to the spinal cord, then the brain can have more time with executive function. Because if we had to think about every single reflex, it'd probably make it worse than better. So, with the deflex strip reaction, as I was saying, patellar tendon gets hit. That will cause for an EPSP or IP. So yeah, it'll get hit. Let me think about it. So yeah, so we'll get hit. It'll cause for a signal to be picked up. Hey, we got hit, and the spinal cord will release, saying for this muscle to relax. So EPSP to relax, and here would be the EPSP to contract. So as we're relaxing, this other muscle is contracting, or our hamstrings are contracting. So that's that. Withdrawal reflex. We're gonna get this guy. He's happy going along. If you were to get a little needle and stick it right into his back, then his arms would move out of the way, away from the source of pain, and his body would, his body would move out of the way, his arms would move this way so he doesn't fall over. And then depending on where you hit him, it'd be the same kind of similar response of, because he's happy about getting stabbed with a needle. So a painful stimulus will trigger an ipsilateral flexor activator and contralateral extensor activation for withdrawal. That's a fancy way of saying what we were saying earlier. So we stab. So ipsilateral flexor activation. So contralateral extensor. So if we're getting stabbed, these move out. Body moves away from the source. Arms move out to move out of it. Or in the back, you move forward. Your body moves forward and your arms move back if you get stabbed in the back. Or the side like that, or like that side like that. So yeah, those are all different reflexes. Let me double check that we covered everything. It's a little bit longer video and it took me to different stuff, but yeah, cool, good stuff.